So without further ado, I'm going to introduce our first presenter, Dr. Michael Bruckman, and he is with Haima Therapeutics. Michael, welcome. You can go ahead and share your screen, and we look forward to hearing all about your company.
Great. Thank you, Katie. And thank you, AngelMD, for this opportunity. So let me get this pulled up. I'm looking forward to getting some good feedback and interacting with some of your network here. All right. Thanks, Katie. Again, I'm Mike Bruckman. I'm the CEO of Haima Therapeutics, and we are a preclinical stage biotech company located in Cleveland, Ohio, in the Midwest. And we are developing a variety of platelet-inspired therapeutics to control bleeding and also to treat other blood-related disorders where platelets play a key role in their progression, such as thrombosis, inflammation, and cancer. Our lead program that I'll be talking about today is an IV injectable hemostatic drug to stop bleeding that can be taken anywhere, meaning this will be a frontline treatment after traumatic injury, saving thousands of lives. Now, traumatic injury or trauma is actually the #1 cause of death in people under the age of 44. And that's because there are no products really available to control non-compressible or truncal bleeding at the scene of injury and during that early transportation stage. The next biggest problem for uncontrolled hemorrhage is in bleeding complications that arise during complex surgeries, such as orthopedic surgeries and spinal surgeries, where there's a high reliance on topical products due to the difficulty of accessing blood products. And finally, the biggest consumers of donor platelet products are people with low platelet count or thrombocytopenia, and they actually consume over 1/3 of the donor platelet supply. Now, why are platelets used? They're used because they're extremely effective at what they do, and that's controlling bleeding and preventing bleeding from occurring. And that's why over 2 million units of platelets are collected and transfused each year. However, there's a number of shortcomings of these donor platelet products that actually lead to over 20% of the collected platelets being thrown away. And that's because of their really strict storage requirements, lack of portability, very short 5-day shelf life, and limited donor supply pool. This has led to the development and use of a variety of other hemostatic agents to treat bleeding. Now, all of these agents have their own major disadvantages, such as extreme cost, inability to affect primary clotting, or for external hemostats, really a lack of the ability to affect systemic bleeding. To this end, a lot of research has been looked into over the past several years into artificial platelets as an alternative therapeutic strategy. Our solution to this problem is a platelet-inspired therapeutic called SynthoPlate that is engineered to amplify the body's natural clotting mechanisms by mimicking platelets' function of sticking to an injury site, then aggregating with locally activated platelets, providing additional touch points like Velcro to strengthen the initial platelet plug. This is achieved through surface decoration of our liposomal technology with 3 unique peptides that drive its function. As a result, we've developed a product that has scalable manufacturing, is sterilizable and shelf stable, can be stored as a powder and reconstituted for use. It has reproducible function, and it can be integrated with the available hemostatic agents to work together to control bleeding. We currently have a license to 4 issued patents that cover all of the composition of matter and methods of use for the synthetic platelet platform technology. Now, you're all scientists and clinicians out there, so to prove its SynthoPlate's function, we first induced a thrombocytopenic defect in mice by removing more than 90% of their circulating platelets, leading to a significant increase in bleeding time. Now, what we've been able to see is a return of hemostatic function with increasing doses of SynthoPlate. In a follow-on study, we actually compared this to the real thing and showed that it's very similar in function, not quite as good, but we show that it's a reproducible hemostatic effect, and we're able to control bleeding and when very few platelets are available. Next, we wanted to test our product in a traumatic injury model. So we actually took pigs, exposed their femoral artery, and took a 3.5 millimeter punch and nearly lacerated the artery. This leads to complete mortality after about 90 minutes. Now, therapeutic treatment with SynthoPlate about 5 minutes after injury, we were able to see significant improvement in survival as a result of reduced blood loss and normalization of arterial pressure. So this is really exciting. This is how we got to where we are today. What I need to point out, though, is that in all of our in vivo and ex vivo studies, there's about 3 or 4 other in vivo models, we were not able to detect any signs of systemic clotting or adverse effects. It's very important for a hemostatic product that's causing clotting. So it's very safe. Similar to other new technologies, the initial research began over a decade ago at Case Western Reserve University, leading to over 7 peer-reviewed publications on its development. This early success led to Haima's formation in 2016 to translate the technology. After I joined in 2018, we were able to secure SBIR funding from NIH, NSF, and DoD. Now, over the last 18 months, we've actually raised an additional $8 million in non-dilutive funding from the DoD and NSF to continue developing and de-risking this lead program. In addition to that, it's actually helped us secure an exclusive license to the technology and build out the team. As a drug development company, we're following an established pathway to market with anticipated value generating pre-IND, IND, and clinical milestones, and the associated costs that you can see along the bottom. Now we're anticipating an anticipated potential exit event after Phase II clinical studies. That's why it only goes out to that point right now. We can talk about Phase III down the road. Now, our intention is to supplement these costs, not just with venture capital money, but also with non-dilutive support from DoD and BARDA, which is consistent with other programs similar to ours. Simultaneously, we're going to seek to form a strategic partnership with an established company for research support, ideally leading to a possible M&A or licensing exit event. To that end, we've actually had multiple conversations with most of the companies listed here and formed agreements with 2 of them already, which really validates our business and fundraising strategy. So far, we've been able to build a strong team of scientists and entrepreneurs to manage the company. We have a kick-ass SAB of clinicians, surgeons, blood banking experts, and biologists to guide our translational experts. And we've engaged a strong team of partners and consultants with unique expertise to push our product into the clinic and beyond. Now, as we move into the clinic, we will be looking for early adopters to provide feedback and evaluation of our product. So anyone in the audience with expertise and experience in complex surgeries or hematology oncology, please feel free to reach out to me at the email below. And with that, I'll hand it back to you, Katie.
Fantastic. Thank you, Michael. This is, clearly you've put together a great team, and this seems like a no-brainer to me, right? This is not something that we have had. It seems better than what is existing on the market and available to physicians when there is either thrombocytopenia or acute bleeding. So very cool. Thank you so much for sharing with us. One of my questions really is around the data that you presented today. So that data was in animal models.
That's right.
How certain are we that this is going to work in humans? And can you talk a little bit about your first in-human studies?
Yeah, absolutely. That's a good question. And it comes up quite often, especially with partners that we have ongoing. So as much research as we've done into platelets function, the binding moieties that our particles are binding to are conserved across species between mice, rats, pigs, and rabbits, and even up to humans. So we're anticipating that the function is going to carry into humans based on the already demonstrated function in mice, rats, and pigs. And our clinical strategy is based on, if we go back to where the problem exists. So traumatic injury is clearly where there's the biggest clinical need or pharmaceutical need, because there's really no strong products for controlling bleeding at the scene of injury and during transportation. However, that's an extremely difficult clinical pathway, and it adds additional unnecessary risk for a first-to-market product. So we've developed a safer and more established clinical pathway where, after safety dose-escalating Phase I studies, we intend to go into high bleeding surgeries such as spinal and orthopedic surgeries or hip replacements, knee replacements, in addition to actively bleeding patients with low platelet counts. So that's how we've kind of, based on the advice of our regulatory council and our SAB, consider the kind of the safest pathway to market.
So there is one question from our audience tonight. Dr. Paul, I'm going to call him. He was asking about how, specifically about the surgical use case, and how, as you mentioned right here, there is a high reliance on topical products in those types of situations. But can you talk a little bit more clinically about how this might work in a clinical situation like a surgical bleed or post-surgical bleed?
Yeah, if a surgical bleed if excess bleeding isn't able to be controlled by so I mean, typically in surgeries, based on the feedback that we've received, donors at risk of bleeding will have some of their blood kind of collected and stored with like a cell saver and then retransfused when the surgeon or anesthesiologist thinks that, you know, they're not able to control the bleeding with a topical glue or gel. But what happens during that process is all the platelets that are actually that would be useful to actually stop the bleeding are more or less destroyed or become dysfunctional through the collection, storage, and retransfusion process. So this would be used in conjunction or as a replacement for specific platelet use, or when there isn't like an interest or need in trying to access blood products to transfuse hemostatic agents to control bleeding. There is a caveat here in the surgical space. We do typically want to stay away from cardiac or cardiovascular surgery and focus more on like the high bleed surgeries. So this is more of an acute single dose treatment to mitigate active bleeding in those cases.
Great. And it sounds like, again, this would be used alongside transfusions of not only probably packed red cells, but platelets, other things that would be currently used for patients in an active bleed, or specifically if a big artery was cut in that scenario. They would be, the surgeon, I'm assuming, would be using all methodologies that are available to them, including your drug.
That's correct. Yeah, and we actually, that's some of the focus of the ongoing grants that we have with the DoD is how our product combines with other blood products to really save traumatically injured animals in this case. And we need to be clear on how this could really be like a force amplifier for transfusion of platelets or plasma or RBCs to be a good complement to current treatment strategies.
Yeah, and another question from our audience. Is there any chance that this would lead to, I'm going to say, overcoagulation, hypercoagulation? Is there a way to overshoot? Are we putting too many things in there?
Yeah, that's a good question. So, during the coagulation process, the clots are being formed and they're also being broken down. So the body kind of naturally is able to break down a clot that's being formed at the injury site as it's growing, so it doesn't grow too big. Because if it grew too big, then we would all clot up and die or have heart attacks and strokes anytime we got a cut.
Right.
And so that's a natural process that the body goes through. And these being lipid nanoparticles will be broken up by lipases. We see no signs that there's a difference in the clot properties that are formed with our particles versus without our particles using some ex vivo assays. And the second aspect of that is the risk of downstream clotting from occurring. And the particles are actually designed to interact only with activated platelets. And platelets only get activated really at the site of injury. And so they're only really interacting with the platelets in a high enough concentration to form a clot at the site of injury. And if they're not interacting, then they'll just get cleared out through the liver and spleen, which is typical of any kind of nanoparticle drug product, with a half-life of 6 to 24 hours. And it's still under investigation.
So I have several more questions. I'm going to go with two that are coming from our audience.
Okay.
And hopefully during the networking, you can talk about other use cases for this technology, because I am guessing there are many. But two questions around what's the shelf life? How is it administered? And someone who's asking, do you need a device for reconstitution? Because maybe there's another startup you could partner with to make that happen.
No, that's a good question. Yeah, you know, we're still very early in the development, so we haven't partnered with anybody to develop a device for reconstitution. It's certainly of military interest, where you have to have something that's self-contained, so that there isn't extra bits and pieces, and it's extremely simple to use at the site of injury when it occurs in the field, or domestically as a car accident victim or gunshot type of use. For reconstitution, we've been able to just mix it with a small volume of water. So the volumes that we're transfusing and that we would be reconstituting are about, you know, 1/10 of the volume of a unit of platelets. So for humans, maybe 50 mils as opposed to 250 mils. So the volume is just maybe 30 seconds to a minute of mixing with water, and it's fully reconstituted. And it's injected intravenously. We're also going to be evaluating its efficacy injected as an intraosseous administration, so you know, through the bone marrow.
Sure.
And I
But
think
it sounds like very stable shelf life as well.
Yes, yeah. These types of products have shelf lives that extend beyond 2 years. So
yeah, and easily reconstituted in the field. Well, Michael, thank you so much for presenting tonight. Very exciting. Haima Therapeutics, again, thank you for being here.